At a high level, QCI Precision Insight provides clinical interpretation of cancer molecular diagnostic tests, and we provide report-ready text and reports back to laboratories that we collaborate with for them to provide to their oncologists that have ordered the diagnostic tests. And I can give you a little bit of history. QCI Precision Insights was originally a company called N of 1 that was formed in 2008. It was founded by a woman named Dr. Jennifer Carter. And her vision was, at the time, there were all these personalized therapies and targeted therapies that were being developed in clinical trials, and they were having variable amounts of success, but a lot of regular patients didn't have access to these therapies, and she wanted to really bring them from the bench to the bedside, to bring these therapies that were available either in clinical trials or very limited knowledge about them, get the knowledge out there to doctors and to patients so that they could be treated. And so I was one of the first employees that was hired back in 2008. And at the time, we worked directly with patients and their physicians to identify, based on their cancer type, what were the most likely targets to look for. So what were the most likely mutations that they might have, and what drugs were available to target those mutations. And so we would compile the information and write reports for the doctors. And as we started to compile that information over time, we accumulated quite a bit of information and started to build up a database. And as we built more and more information, we began collaborating with diagnostic companies that were actually doing the testing to provide the information and reporting for their reports. And so one of our early collaborators was Foundation Medicine. And when they launched their Foundation 1 panel, all of the interpretation at that time for the first 2.5 years was done by N of 1. The work is done by a team of PhD scientists. They are handpicked and highly trained to a detailed curation protocol that we have developed over the years. And they curate according to this protocol. And so all of the information is very standardized. All of the information that's clinically related is reviewed by our team of oncologists. So it's a very high quality, highly reviewed product that goes onto these reports. One of the things is the structure of the reports. So they are all structured very, very similarly so that the physicians always know where to look for the information they want. So if they want information about the specific mutation in terms of the biochemistry, they know to look in the molecular function section. And if they want details about what were the results of a clinical trial for a particular drug, they know to go to the clinical evidence. It's very clear from report to report and biomarker to biomarker where that information is in every single report. It's always standardized. They also know that the quality of the reports is very uniform, that we have this very highly trained team of PhDs, that all of the clinical data is vetted by oncologists who are practicing and very familiar with all of these therapies and targets. The other thing that makes it stand out is, frankly, the turnaround time and the breadth of coverage that we have for these biomarkers. Because any lab that was trying to build up this information, it would take them years to get the level of coverage that we have, which we can return in 2 minutes. So the laboratories that we collaborate with, in the beginning, they were doing individual gene assays, but now at this point, people are doing much larger tests, like 50 gene panels, 100 gene, 200 gene, 300 gene panels. And the labs that we tend to collaborate with have their own bioinformatics pipeline. They have a very robust filtering mechanism so that they can do the DNA sequencing and then identify the most likely mutations that are going to be clinically relevant and send us a refined filtered list to QCI Precision Insights, along with a diagnosis and a little bit of demographic information about the patient in terms of their age, their gender, and the broad location. So no protected health information is transferred to us, but just enough to be able to do some good clinical trial matching. And so that information comes into our system, and then our team assembles the reports with the help of a database and software system, and we return the reports, and then the company that we're working with takes the information either in the form of a PDF or in the form of a structured data file that they can create their own branded report. So the labs we work with tend to be, fairly high throughput. They are looking for a solution that will give them economies of scale, because they are doing all of the sequencing, the wet lab, the bioinformatic work, but they don't want to hire a whole team to do all of the literature searching and the drug matching and the writing of these reports. If they can send it to us and get that information back and be able to quickly review and sign it out, they get those economies of scale. We've built a very large database. We've curated over 1,000 cancer genes, well over 500,000, approaching 750,000 variants across nearly 1,000 different cancer subtypes. And so that information is organized in a hierarchy. It's very standardized and it's reviewed regularly so that we keep a certain amount of that information up to date based on how often we see it. And so if we're keeping a certain body of that information always up to date, so that when a case is submitted to us, our system interrogates the case and says, okay, which of these variants has the system seen before, which have not been seen before? And if everything in the case is complete, so for example, it's a KRAS mutation in a lung cancer and a TP53 mutation in that same case, both common, both seen before, the system will assemble the report and send it right back out. And it'll go back to the laboratory within a minute or two. If it's a bigger case with more uncommon genes or uncommon variants, or something it's reached its expiration and needs to be looked at by a person, then it goes into the work queue for our team of scientists. And our team of scientists pick up those cases and work through them, reviewing the variant and assessing the role of that gene in that disease, any drugs, levels of evidence for those drugs, and assembles the report and then sends it back. And so on average, the cases that do need human attention, we contract up to 2 days for the turnaround time based on the size of the panel. But in reality, they're typically returned on average within half a day and sometimes only within an hour or two. So the turnaround time is pretty quick. It's from the scientific and medical literature. Also, databases like Cosmic and ClinVar. So our scientists who are writing these expert interpretations have a protocol that they follow. We have a system that pulls a lot of information and presents it to the scientists so that they are able to look at everything in one place without having, 17 windows open. But they do also go out to Google and PubMed and really scour the internet for information about that variant. So they're pulling in all of this information, reviewing all of the papers. They dive into the supplemental data. They look at the figures. They evaluate the experiments. They evaluate the controls. They really want to get a very good sense of what is the available data that supports this variant. So if we think it might be an activating or targetable variant, what's the data? Is it, has it been seen in people? Has it been treated with a particular drug where there was a response seen in a person with this disease? Has it been studied in cell lines where it was shown to drive oncogenesis? So all of these pieces of information are taken together, and then the scientist reviews all that information and writes a short summary with references so that the physician or the lab director can review that information and make the determination, do I agree with this information? And they, over time, build trust because they know that everything we return is well researched and well balanced. In a lot of cases, what we see is variants that sit right on the edge, where there might be one piece of information that suggests that it's a clinically actionable variant, and another piece of information that suggests it isn't a clinically actionable variant, or the data using sort of a modified version of the ACMG guidelines or the new ClinGen Vic guidelines, where it sits right at the edge of VUS or clinically actionable. And as scientists, we have to make a determination one way or the other, and we try to find as many criteria as we can. And then, of course, there are variants that don't fit any of the criteria, and we have to make those decisions. But because there are human beings that are very highly trained and knowledgeable, they'll apply the knowledge that they have and make those decisions in as consistent a manner as possible so that our clients can always rely upon, like, what information are we using to make those decisions. So we do have two main ways that people like to use the service. In some cases, our clients want to see interpretation for every single variant in the case. They don't want anything to be skipped. They want a full report of information. In other cases, clients will say, well, your coverage is so good. You have so many variants in your system. I really want my report back right away. And I'm going to take whatever information you have at that moment, as long as you also tell me what you didn't look up. So what we would do is our system would assess each variant in the case. This happens within a second that it's going through and assessing each variant and determining whether it's been seen in our system before and whether it's up to date. In the case of these automated or pre-curated cases, the system will go through, and if there's a variant that our team has not seen before, it'll report out variant not curated so that the lab director knows it's not that there's no information about this variant, it's that our team hasn't looked. So if you want to look for that, if you want us to go and fill in the blanks, we allow a reflex. So they can send the case back and say, please, in code automatically, please reflex this case for us. And then it'll go into our queue and we'll fully curate it. But a lot of the time, the groups that are getting those cases back will look and they'll see what wasn't curated and they'll look at it and they'll say, this is probably a VUS anyway. It's not targetable. We don't need to wait for the full curation on that report. I can talk through an example that we have. So this example is a case of acute myeloid leukemia, or AML, and it's a case that has a FLT3 mutation and an NPM1 mutation. So it's a small case. So what information do we show for an individual biomarker? The first section will be a high-level summary. What does this variant do? Are there any drugs that would be associated with it? Does it have any prognostic or diagnostic significance? So at a very high level, what are the takeaways? What do I need to do about this variant? And then each section gets progressively more detailed. We have a section on biological relevance where we'll give a detailed description of the molecular function of that variant. So with the FLT3 internal tandem duplication, what part of the protein did they occur in, like the juxtamembrane domain? Has this variant been shown to be associated specifically with drug response? And what are the references? Every sentence in the report has detailed references with the PubMed IDs. We'll tell you how often are these mutations seen in this specific disease type. So, FLT3 mutations reported in 23% of AML cases in COSMIC. And so we're pulling that information from COSMIC, from CBioPortal, and also from the literature. And then the next section gets into the clinical relevance. What role does it play in disease? Is there any prognostic or diagnostic significance for this variant? And what is the overall effect on drug sensitivity? Again, with the references, what drugs are available, and what's the data to support that at a high level. So the final and most detailed information we give is this detailed clinical evidence, where we'll give summaries of the clinical trials that led to the approvals of the drugs. So, for example, with FLT3 AML, we'll give a 1 or 2 sentence summary of the phase 3 clinical trial of midostaurin in FLT3 mutant AML patients. And this information is specific to the disease that the patient has, so AML, and it's organized by the phase of the clinical trial from which the data is derived. So phase 3, phase 2, phase 1. And that is about it. Our reports also will give a summary list of sample therapies as well as matched clinical trials that are ordered based on both the geography of the patient, if that's provided, as well as the clinical specificity of the trial for that patient.
QIAGEN. Sample to insight.